Hello everyone. I hope you all can see the screen well. Assalamu alaikum and good afternoon everyone. I am Abeduzzaman Anon and I have completed my undergraduate studies in pharmacy from IUB and I am currently working as a research assistant at Bioinformatics. So today I will be reviewing a recent epigenetic focused bioinformatics study on Alzheimer disease titled Identifying the Function of Methylated Genes in Alzheimer disease to determine epigenetic signatures, a comprehensive bioinformatic analysis by Rezanur Rahman. Well, he is my mentor. He taught me everything about network pharmacology and everything. Well, thanks to him and his colleague for publishing in experimental result in 2021. So this journal was published in Cambridge University Press, and as he was my previous mentor who helped me learn network pharmacology and helped me complete several projects. So I'm really proud to present this article today in this platform. So let's start. If I talk about the introduction of Alzheimer disease, I will say Alzheimer disease is the most common form of dementia, and it marked by progressive cognitive decline and memory loss. Pathologically speaking, it is characterized by the accumulation of amyloid beta plaques and neurofibrillary tangles in the brain. While classic mechanisms of Alzheimer's disease involve protein aggregation, recent research shows that genetic and epigenetic factors also play a crucial role. Epigenetic means modifications in gene activity without changes to the DNA sequence, and one of the main mechanisms is DNA methylation. It is a chemical tagging of DNA that usually suppress gene expression. Well, in this study, the author specifically integrate gene expression and DNA methylation data all together to uncover epigenetic signatures associated with Alzheimer's disease. And the primary objective of this study was to identify epigenetic signatures associated with Alzheimer's disease by integrating gene expression and DNA methylation data. So why combine gene expression and methylation data? Most previous studies examined gene expression or methylation separately. This study's major target was to combine the two, mRNA GSE4757 from the NCBI GEO dataset tells us which genes are turned on or off, and DNA methylation dataset GSE45775. This dataset tells us where methylation might be controlling the expression. So in this diagram, we can see the code from where these databases were taken, and they all are available in NCBI GEO dataset. So this dual analysis allows the author to identify methylated differentially expressed genes that are both methylation altered and differentially expressed in Alzheimer's disease and control brain tissues. So let's talk about this article's study methods. A simplified step-by-step walkthrough of what the research did in this article, I will discuss about this. In data collection, they used publicly available datasets of post-mortem brain tissue from Alzheimer patients and matched them with controls. Gene expression analysis was performed using GEO2R and an online platform. This is an online platform for identifying differentially expressed genes. That means DEGS. And DNA methylation analysis was used to identify differentially methylated genes. That means DMGS. And the threshold value +2 greater than +2 and less than -2 was considered as the cutoff criteria. Here the t value represents t statistic. That means that measures the size of the difference relative to the data's variation. In simple terms, it tells you how many standard errors the observed difference is away from zero or null hypothesis. A large absolute p-value, positive or negative, means the difference between group is large and the signal is strong relative to the noise. A small p-value means the difference is small and the result may be due to random variation. So this t-value is calculated in t-test, which compares the means of two groups, disease versus control. And p-value represent the probability of observing your result or something more extreme if the null hypothesis is true. The null hypothesis usually states there is no difference between groups. So the p-value represent if there is actually no real effect or how likely it is that we would see this result just by chance. Small p-value less than 0.05 means result is statistically significant, and large p-value greater than 0.05 means result is not statistically significant. So a p-value doesn't measure effect size. It only measures statistical evidence against null hypothesis. Then they found the overlap between differential expressed genes and differentially methylated genes. These are methylated differentially expressed genes. They categorized these into hypermethylated, low expressed genes, that means hyper-LGs, and hypomethylated, high expressed genes, that means hypo-HGs. Enrichment analysis and network analysis were done through pathway enrichment to see what biological process these genes are involved in, and protein-protein interaction network to find key hub genes. Additionally, transcription factor analysis was conducted too to explore upstream regulatory elements that could influence the expression of these genes. So now we will discuss about key results of methylated differential expressed genes and their functions. In the methylated DNA differentially expressed genes, the analysis identified 28 methylated differentially expressed genes associated with Alzheimer's disease. And 18 of them were hyper-LGs and 10 of them were hypo-HGs. That means they are hypermethylated, low expressed genes, that is suppressed genes. These genes are suggesting transcriptional repression. They are lowly expressed and these were largely involved in cell signaling and metabolism pathways, including the neuroactive ligand receptor interaction pathway, a pathway important for neuron communication. And for hypomethylated highly expressed genes, they are activated and they suggest less suppression, high expression. These genes were enriched in pathways related to transcriptional regulation and development, especially in hippo signaling pathways that we can see from here. And their roles are in cell growth and neural development. The KEGG pathway enrichment analysis was performed to identify the biological signaling pathways associated with the methylation-driven genes. The results showed distinct pathway patterns between the two gene groups, hyper and hypo, suggesting different regulatory mechanism in Alzheimer's disease. For hypermethylated low expression genes, that means this side, these genes were enriched in pathways such as nitrogen metabolism, circadian rhythm, pentose and glucuronate interconventions, nicotine addiction, etc. Nitrogen metabolism may reflect altered metabolic process in the brain, which is important because metabolic imbalance is known feature of neurodegeneration. Circadian rhythm is particularly relevant as sleep disturbances and disrupted biological clocks are common in Alzheimer's disease and they may contribute to disease progression. Pentose and glucuronate interconversions are related to cellular metabolism and detoxification process. Nicotine addiction pathway does not mean addiction itself, but it includes neurotransmitter-related signaling components that are involved in neural communication. Overall, these pathways suggest that hypermethylation may suppress genes involved in metabolic regulation and neural signaling. For hypo-HGs or hypomethylated high expression genes, these genes were enriched in pathways such as HIPPO signaling pathway, CGMP PKG signaling pathway, alcoholism related pathway, viral carcinogenesis, RNA polymerase activity, etc. The HIPPO signaling pathway regulates cell growth, survival, and development. Its dysregulation may affect neural maintenance and brain structure. The CGMP PKG signaling pathway is involved in cellular signaling and synaptic function, which are essential for learning and memory. Some pathways like viral carcinogenesis and alcoholism include genes involved in cellular stress, inflammation, and signaling network. These doesn't mean that the gene is cancer or caused by alcohol, but the shared molecular components overlap with these pathways. After that, hub genes and network connectivity identification was conducted. By constructing protein-protein interaction network, the authors identified several hub genes, genes that interact with many other and could be central to disease mechanism. Among hypermethylated low-expressed genes, as you can see, TOMM22, TBX5, ANK2, GRIA2, these genes are mentioned. And among hypomethylated high-expressed genes, BMP2, GATA4, HDAC11, these genes are highlighted. These genes may be critical players in the molecular network of Alzheimer's disease pathology, as they are mentioned as hub genes. Now let's talk about transcription factor linked to DNA methylated differential express genes. Transcription factors are regulatory proteins that control gene expression by binding to specific DNA sequences. In this study, transcription factor analysis or TF analysis was performed to identify upstream regulators of the methylation-driven differential express genes. This helps us understand not only which genes are altered, but also what may be controlling those genes or those changes. The researchers identified transcription factors proteins that help turn genes on or off. They are likely influencing these methylated differential express genes linked to hypermethylated low expression genes such as ELF4, EBP1, XBP1. As mentioned, you can see this. The transcription factors associated with hyper expressed low expressed genes, hyper-LGs, hypermethylated low expressed genes. These transcription factors are involved in cell survival and stress response, such as XBP1 that is linked to endoplasmic reticulum stress. Since these genes are hypermethylated and downregulated, it suggests that methylation may suppress important regulatory networks involved in neural function, immune response, and cellular homeostasis in Alzheimer's disease. Hypomethylated high-expressed genes, that means TFA, P2C, HINFP, SP1, SP3, such as SP1 is a widely acting transcription factor, cell cycle and chromatin regulation, hormonal and nuclear developmental gene regulation. These genes are hypomethylated and upregulated. This suggests increased transcriptional activity in certain regulatory pathways. This may contribute to altered signaling, cellular stress response, or neurodegenerative mechanisms in Alzheimer's disease. So these transcription factors could serve as upstream regulators and might offer targets for further investigation in Alzheimer's disease. So what is biological significance in this study? Let's discuss about that. This integrated methylation expression approach serves several purposes. One, it identifies potential biological markers, genes whose methylation and expression patterns indicate Alzheimer's disease presence. It also reveals epigenetically regulated pathways that is important in Alzheimer's disease pathogenesis. Also, it suggests potential therapeutic targets because altering methylation patterns could theoretically influence gene expression and disease progression. So this is especially important since epigenetic marks like methylation can influenced by environmental factors such as lifestyle or even drugs. Clinical and research implications like diagnostic, some of the identified transcription factors and methylation patterns may be detectable in body fluids, which could lead to early biomarkers for Alzheimer's disease. In case of therapeutics, if methylation pathways are implicated in driving disease, epigenetic drug agents that modify DNA methylation, so these could potentially be repurposed or developed for Alzheimer's disease. Next step for research, as the author suggests, that experimental validation of these methylated differentially expressed genes in vitro or in vivo models. Investigation of whether these methylation changes occur early in disease progression and understanding how these epigenetic signatures interact with known Alzheimer's disease factors like amyloid and tau. So these are the next step the author suggested that they should do. While the bioinformatic approach is powerful, it also has limitations, obviously. Public dataset might contain variability from different platforms or populations. Correlation between methylation and expression doesn't necessarily imply causation, and these predictions require experimental validation to confirm biological relevance. So these are the things we should consider in our research too. To summarize this study, as conclusion, it integrates gene expression and methylation data to uncover epigenetic signatures linked to Alzheimer's disease. It also identifies 28 methylated differential expressed genes as it connects these genes to biological pathways and regulatory networks involved in Alzheimer's disease. It also highlights promising targets for further research in biomarkers and therapeutics. So this study is a strong example of how bioinformatics and epigenetics can be combined to deepen our understanding of complex neurological disease such as Alzheimer's. So that's all from my part. And if you guys have any questions, I am honored to answer those. Thank you.